Topic: Protection and use of animal and plant resources. The value of plants in natural and human life. Number of our students. Key issues. Topic plan. First, protection and use of animal and plant resources. Second, the value of plants in the natural and human life. Third, forest is the most important plant resource. The purpose of the lecture: the study of protection and use of animal and plant resources, the value of plants in natural and human life.
Plants are the most important organisms on this planet. They occupy the bottommost part of the food chain. They are often referred to as producers, and all other organisms are directly or indirectly dependent on plants. Almost 85% of the world's food consumed by humans is plant-based. The plants grown for food are called food crops. Food crops are highly dependent on water availability, soil, and climatic conditions. Sometimes, due to environmental conditions like drought, the crops do not produce sufficient food or the crops fail. This leads to acute shortage of food in that region. It is called as famine. In the year 1943, the Great Bengal famine is estimated to have caused the deaths of up to 4 million people. Also, as the population increases, food production must also increase. And if the food production does not increase, it will cause food crisis. As the land for agriculture is very limited, it is necessary to improve the productivity of the crops per acre. Previously, we have learnt that by implementing animal breeding, we were able to improve the quality and productivity of farm animals. Similarly, by plant breeding, it is also possible to improve the quality and productivity of the food crops. Plant breeding is defined as science of changing the genetic traits of plants in order to create desired plant types that are better suited for cultivation, gives better yields, and have good resistance to diseases. Plant breeding is not a new technology. It is a very old one. Earlier, man used to deliberately crossbreed closely related plant varieties to produce a new plant of desired trait. Nowadays, breeders try to incorporate the following desirable traits in an improved variety: increased crop yield, improved quality, increased tolerance to environmental stresses like salinity, extreme temperatures, drought, and frost, increased resistance to pathogens, increased tolerance to insect pests. This process of breeding closely related individuals that are genetically different to produce a hybrid offspring is known as hybridization. Scientists Gregor Mendel and Charles Darwin effectively demonstrated hybridization within a plant species. After that, the process was further developed by many plant breeders. Plant breeding is a systematic process. The entire process of plant breeding, starting from choosing desirable qualities of plant to the final product, is broadly divided into 5 main steps. Let us learn about them one by one. Step 1. Collection of variability. We know that plant of particular species will have different varieties on different parts of the planet. Each variety will have few different sets of traits that are different from each other. This is known as genetic variation. For example, ragi. There are about total 121 natural variants of ragi on this planet. Every one of them is different from each other by few traits. Some show disease resistance to particular bug and show drought resistance. Some give high yield and no resistance to drought. Some give less yield and show drought resistance, etc. All these are desirable characters. So to create a new variety of ragi with better characteristics, it would be better if one knows all the natural varieties of ragi available on this planet. From that, it would be easy to select the best required traits for the new plant. From the year 1971, under the guidance of International Board of Plant Genetic Resources, IBPGR, several institutes have started collecting all the naturally available plants and seeds, having all diverse alleles for all genes in a given crop from different geographic locations on this planet, and made a library out of it. This library is called as germplasm collection. This forms a resource from which new varieties are created. Currently, most of the crops have germplasm with more than 100 varieties. Due to availability of vast germplasm, any breeder can now easily manipulate different combination of alleles in order to create a superior plant. There are 2 germplasm collection centers in India. These are International Crops Research Institute for Semi-Arid Tropics, ICRISAT, at Hyderabad, and National Bureau of Plant Genetic Resources, NBPGR, at New Delhi. Step 2. Evaluation and selection of parents. When varieties were first collected, it was completely based on easily distinguishable characters. However, plants' variations are due to the environmental conditions present at that time, which is temporary. For example, suppose researchers collected a dwarf plant from drought region and named it as dwarf plant. But when this variety was cultivated under normal environmental conditions, the plant grew tall. It means that the dwarfness was not actually the trait of the plant. It was just a result of environmental conditions. In order to avoid such type of errors, careful evaluation and characterization is required to know about the composition of the germplasm and the diversity of the given crop. Let us learn how the plants are evaluated and characterized. Before the plants are evaluated, the plants are tested for pureness. If a plant has more than one type of allele for a particular trait, it is called heterozygous. Such plants are rejected, and only pure plants or homozygous plants are subjected to further evaluation. Now, let's learn about plant evaluation. First, all the samples are grown in the field under controlled environmental conditions with good protection. Then, it is made sure that the plants completely adapt to the place they are grown. This ensures that all their traits are completely expressed. Once they are adapted, the plants are tested under various stressful conditions such as pathogens, salinity, drought, etc. All the responses are recorded. Apart from this, the plants are also tested in laboratory for different molecular and biochemical characteristics. Once all the enzymes and the biochemicals produced are known, the information is neatly recorded and labeled on the plant. Using this wide range of information, scientists can easily select plants for breeding. The selected plants are later inbred or self-pollinated to obtain pure copies of the homozygous parents. Cross-hybridization among the selected parents. Now, since we have selected pure line parents with desired characteristics, the parents are now cross-hybridized. Cross-hybridization helps to produce hybrid progeny. This helps to bring desired traits from different plant lines into one plant line. For example, if we cross a low-yielding, high disease-resistant ragi plant with high-yielding, low disease resistance ragi plant, since the parents that are crossed are pure line, the F1 hybrid obtained will have superior characters of both the parents. The progeny here will be high-yielding and high disease-resistant ragi plant. This tendency of hybrids to obtain superior characteristics of both parents is known as heterosis or hybrid vigor. The term was coined by Schull in 1912. However, heterosis was first studied by Carl Reuter in 1763, followed by Darwin in 1876. Let us understand how cross-hybridization is done by breeders. It is done by a process called artificial hybridization. Artificial hybridization is the production of hybrids by cross-pollinating two separate plants under controlled conditions under the supervision of the plant breeder. This process of artificial hybridization in plants involves various steps. These are as follows. Selection of parents with desired characters. Care is taken while selecting parents for hybridization program. All the desired characters should be present in parents which are required in the new crop variety. Emasculation. Anthers or male parts of the flowers are removed. Now flower contains only female parts. This is done to prevent self-pollination among flowers. Bagging and tagging. Soon after emasculation, the carpels are covered polythene bags to prevent contamination of stigma by foreign pollen. Collection of pollen grains. At maturity, the pollen grains are collected from desired parent plant in dry plastic bags. Artificial pollination. When the stigma of the emasculated flower matures, the polythene bag is removed, and with the help of a brush, fresh pollen from selected plants is dusted over the receptive stigma of the female. Tagging. A tag is with relevant information attached to the plant. The tag bears the information like date of emasculation, date of crossing, and detail of male and female parents, etc. Desirable combination of variations. Seeds obtained after crossing are sown and evaluated for desirable and undesirable traits and are screened properly. Back crossing. If desired traits are absent in the progeny, then they are back crossed again till the desired traits are obtained. Limitations. Cross hybridization is a very time-consuming and tedious process. The cross hybrids may not combine the desirable characters. Usually, only 1 in few hundred to 1,000 crosses show the desirable combination. Step 4. Selection and testing of superior recombinants. The hybrid obtained is scientifically checked for desired characters. If the test is passed, the plants are self-pollinated for several generations. Since the F1 hybrid has both dominant as well as recessive alleles, at every next generation, we will get wide variety of combinations. Some will be desirable and some will be undesirable. At each generation, the undesirable plants are eliminated, and the process is continued for 7 to 8 generations until uniformity or homozygosity is obtained. This will create a pure line where characters do not segregate. This pure line is called as cultivar. Cultivar is plant with desirable characters that can be maintained by propagation. To evaluate these cultivars, the cultivars are grown in research fields and their performance is recorded under ideal fertilizer application, irrigation, and other crop management practices. In India, Indian Council of Agricultural Research, ICAR at New Delhi, carries out these evaluations. After evaluations, the plants are again tested in farmers' fields for at least 3 growing seasons. The testing is done at several locations throughout the country, representing all the agroclimatic zones where these crops are usually grown. Using the test results, the cultivars are evaluated by comparing it with the best available local crop cultivars in the farms. If the new cultivar is far better than the existing cultivar, it is selected for seed multiplication. Step 5. Multiplication of improved seed. The seeds of the approved cultivars are multiplied by growing them in suitable conditions. A large quantity of seeds is obtained. The seed are then tested for quality and certified by National Seed Corporation, NSC. Such tested seeds are called as certified seeds. Now the seeds can be distributed to farmers and licensed seed sellers.
Answer the test questions in the online class. Watch the video on the YouTube and send task to the university system. Thank you for your attention.